Um, what I'm going to talk about is a recent study that we at Schrödinger have undertaken, which is, I would say, not quite AI4Proteins, but I think is a very related topic that should be relevant for everybody who is building protein structure models by any type of approach, because it is about validating those structure models for further computational or other insights. And maybe just to start, let me state the problem that we're looking at here. It's probably not controversial to say that the gold standard to determine what a protein or a protein ligand complex structure looks like remains protein crystallography. That is, especially for somebody like me who comes from a structure-based drug discovery software company, that is the step that we care about a lot, because if somebody gives us, and my GIFs should also be moving. Hope you can see that as well. If somebody gives you this nicely highly resolved structure where every single side chain is easily identifiable and placeable, this is where a lot of the computational tools that Schrödinger is developing can be brought to bear quite productively in a drug discovery or similar study. However, as of course also everybody knows, and that's probably part of the reason why most of you are here, it's not always possible or cost effective to generate a high resolution X-ray structure for every molecule that you care about. There are alternative ways to obtain 3D models, which I hope is also uncontroversial to say, will usually be of a lower quality than high resolution X-ray structure. And these can come from full de novo prediction, they can come from homology modeling, from cryoEM or other structure determination methodologies, and so on. Having such a structure can still be extremely valuable for any project, but I would say they come with a need for additional validation to make sure that, for example, structure-based computational methods are really applicable based on the structure that you have. So we're talking about the question, can I understand molecules binding to a protein binding site which might not be perfectly resolved or might not be quite optimal for the molecule binding there? And the way to understand or to study this question, we realized that this is quite a similar problem statement to induced fit or cross-docking. And a quick reminder, as probably all of you are very aware of, ligand docking calculations usually use rigid receptor and flexible ligand structures to generate possible bound poses. This works quite well if the receptor structure is pre-optimized towards the kind of molecules that are binding, either because they come co-crystallized with a similar series or because the receptor is generally not changing very much. It becomes a lot more difficult if there are induced fit effects. So if the receptor is adapting to the different molecules, then placing them with a rigid receptor model is no longer all that easy. And some kind of consideration of induced fit effects needs to be done to generate proper poses. Again, kind of a similar problem. Can I place a molecule into a structure that is not optimally arranged for that molecule to bind in it? And since induced fit effects can, of course, span a giant range of possible conformational changes, we'll limit the exact problem statement a little bit further to structures that have side chain rearrangements. We're not talking about DFG in, DFG out changes, massive conformational changes of a protein, but small changes. And things like the one that you're seeing here. I have two X-ray structures of thrombin receptor. You can see that they are, by terms of backbone, quite similar to each other. But the white one allows me to easily dock this known molecule in its correct bound pose. The green one does not, and the green one does not because really there's this one glutamic acid side chain which is a little bit rearranged, and that is blocking the ligand from entering here. Now, looking at the structure, I can convince myself that it's not that hard to move that glutamate out of the way and make the ligand fit into the green structure as well. And this kind of gut feeling or this identification of residues that could probably be moved out of the way, side chain rearrangements to be encountered, to formulate this into a reliable and predictive algorithm is the challenge for an induced fit docking model. And we have developed a very advanced such induced fit docking model called Ifdmd, and I'm going to talk about how such a workflow might look like as a possible solution of the problem, placing structures into okayish protein, placing ligands into okayish protein structures. We start by surveying the state of the art. How well does this cross-docking work with tools that are available right now? For that, we assembled a series of cross-docks. So structures from, in this case, I think the PDB, where different and not too similar molecules were bound to the same target structure, where there is some visible induced fit effect. So at least one side chain occupies a significantly different rotameric state in the two co-crystal structures, but no major backbone changes have happened. For reasons that will become apparent later, we have split those up into a training or test set. But the first thing we did here was to test both our classic rigid receptor docking using GLIDE and our standard IFD induced fit docking approach, which places molecules and rearranges the side chains around them a little bit. And if we wonder, how well does it work to place, let's say that the top pose comes out, I would say approximately right. Our criteria here is 2.5 angstrom heavy atom RMSD for the ligand molecule that is placed. Rigid receptor docking has about a 1 in 3 chance to give you a pose that fulfills that criteria. Induced fit docking, as it is implemented right now, is about 50-50 in giving you that. And if I relax my consideration and say I want one of the top 2 poses to be in good agreement to the known bound pose of the ligand, then both methods get about 10 percentage points better. So clearly, some promise. Docking methodologies are capable of finding the right poses, but with, I would argue, insufficient performance to be truly reliable. Even if it's 2 out of 3, that's probably not a good enough structure to base a whole lot of additional work on without further validating it. So how could this further validation look like? I'm now going to describe the Ifdmd workflow, and that needs to do 2 things well if it wants to solve the induced fit docking problem. The first one is the sampling problem. I need to generate enough 3D models of my ligand binding to that receptor with appropriately rearranged receptor binding pocket so that the true structure is part of that. And then the second one is, out of this ensemble of generated structures, I need to pick the right one, which is a scoring problem. I need to rank those bound poses according to the likelihood of being the actual correct one. And there's quite a bit of additional effort in both of these areas in that workflow. I will start with the question of sampling improvements. That is, of course, goes to the core of what induced fit docking is like. I need to decide which side chains in the receptor I want to move. I cannot move all of them, of course, because that leads to the well-known combinatorial explosion. Even if I have only a handful of residues that can adopt 3 to 5 rotamers, I'm very quickly getting into millions or billions of receptor structures that I would have to evaluate. For every single docked pose of every single molecule that I care about, this very quickly becomes too expensive. So we have to limit ourselves to a couple of residues that are most likely the most important ones to rearrangements to make them flexible. There's a lot of ways that one can do that. You can base it on experimental data like crystallographic B factors. You can base it on flexibility in MD. You can base it on the amount of solvent exposure that a residue has. None of these approaches were 100% convincing when we looked into them. So the first big change in the Ifdmd workflow is to think about this problem a little bit differently. We need to identify receptor structural changes. Those are induced by the ligand. So we need to place the ligand first to identify the clashes that need to be resolved. But we can't place the ligand without moving those clashes. So we try to decouple those 2 processes by first docking the ligands without being aware of the receptor. And the key point here is that the initial ligand placements happen based on an existing template structure. It could be a co-crystallized one or another known one. And the molecules are placed based on a pharmacophore overlap with that template ligand only, so not aware of the receptor. Since I only need 3 pharmacophore features to generate a possible pose here, this very quickly generates many, many potential ligand-bound poses. So thousands of these are generated. Of course, most of them will strongly clash with the receptor because they are placed without consideration of the receptor there. If they clash with the backbone a lot, we would discard them. But any of these pharmacophorically placed ligands that look like they could be refined to fit into the pocket undergo protein sidechain refinement now. You can see here, if there's only a few clashes, sometimes they can be resolved. That leaves us with now thousands of individual protein ligand complexes, each one with a slightly changed receptor. You can see we're not changing the backbone all that much. It undergoes maybe a little bit of minimization, but the side chains can change quite rapidly. So each of these ligands enforces a more beneficial receptor conformation for itself, and then it gets redocked into this new receptor conformation with standard rigid docking. So this is the part for the sampling, which generates a lot of sometimes very novel and very diverse protein ligand complexes. Now I need to pick the ones that I consider as best representatives of the actual bound pose. And that can be truly difficult because in a way, a standard ligand scoring function only has to account for changes in the ligand, which are usually quite small. Once I start changing receptor side chains, a lot more difficult energetical terms, entropy compensation, solvation effects, and so on happen. So I need to find a way that would allow me to identify, well, this ligand, the green one here, looks like it is placed okay, but it has displaced a side chain here and formed a new interaction that would not actually be stable in a true complex. This is a little bit too much for a simple empirical scoring function to figure out. So now we're moving to molecular dynamics-based ways to identify which of these poses that were predicted are actually stable or maybe less stable. The first step to do here is metadynamics. I'm not sure if you're familiar with metadynamics. It's a biased way to run molecular dynamic simulations, which pushes the system with a biasing force along a defined collective variable. What it effectively just does is it makes the system more likely to explore bigger conformational spaces. It pushes it away from its starting conformation. And in the context of a ligand bound to a protein, what it does, it tries to nudge the ligand to unbind. We're not doing a complete unbinding simulation. We're instead using this metadynamics on the bound poses to see how deep is the well that ligand is in. Is it easy to push it out? Is it easy to disrupt a lot of the interactions that it has formed? And we have seen that this is usually harder for native bound conformations than non-native ones. So we can see it's easier to disrupt them. That is an indication that the pose is not quite stable. Of course, this will not work if you're starting with a metastable pose. Quite often you can have your ligand, let's say, switched 180 degrees around in the binding pocket, still tightly fitting and not easy to dislodge. So the second case would have to be, how do we identify metastable states? And metastable typically means that some kind of hydrogen bond donor acceptor group, for example, in the receptor, is not being satisfied. So we'd have to identify this ligand pose here might look good, might fit well into the binding pocket, but it comes at a huge energetic cost because it is desolvating a polar group in the receptor here. This can be done with appropriate solvation-desolvation penalty terms. So there's MMGBSA calculations that can do that. There is the W-score scoring function that is highly trained to identify such unsatisfied hydrogen bond donors and acceptors. But to properly do that, we need one additional step, which is to predict the water structure within the binding pocket as well, because the water structure will respond a lot to small changes in the receptor. It will respond a lot to how exactly the ligand is being placed. And once those complexes are being generated, we generate possible water structures using, again, molecular dynamic simulations, this time with grand canonical Monte Carlo water sampling and the water map technology to identify where the clusters of waters are. These can alleviate a lot of these buried polar group penalties, but not all of them. And that is an additional part. Clearly, I went through quite a lot of terms in a short time. The scoring function is kind of complicated. You can see it includes molecular mechanics terms. It includes standard docking scoring function terms, including the additional desolvation penalties, contribution for metadynamic stability, and a couple of additional parameters that identify unrealistically large side chain motion penalties. A couple of parameters in that scoring function were optimized. This is where the test and training sets that I mentioned before come in. And how precisely this is done, of course, I have a hard time explaining a 40-page paper in detail. It was recently published, I think, in JCTC. If you want more details about the algorithm, they are available here. What is also hopefully noticeable is that this workflow really combines a lot of already existing technology. Yeah, there's MD simulations in there, there's water analysis, there's binding pocket analysis, there's an existing scoring function, actually two from the guide NW score methods, there's pharmacophore alignment, there's protein refinement, and all of these together build the entire workflow that does an induced fit docking or an induced fit docking-like calculation for us. You can also see that this is a fairly costly calculation